Hello, my name is Mr. Chipman, and I teach biology and AP biology at Murray High School in Murray, Kentucky. This is 3.5 Cellular Respiration, Part 1. We're going to be dividing 3.5 Cellular Respiration into two parts because it's a lot. It's a lot of things, and we want to make sure that we get all the things and do them correctly. And so in this video, we're going to have a kind of an overview of all of the processes. And in the next video, we're going to be looking specifically at the electron transport chain and some of the specificities of the electron transport chain and some of the things associated with that. Let's talk about cellular respiration. So cellular respiration is, first of all, it happens in all life forms. Prokaryotes, eukaryotes all do cellular respiration or fermentation, and they're all doing the first step of cellular respiration for sure, which is glycolysis. We're going to get there in a moment. And cellular respiration is essentially breaking down glucose to get ATP. There's going to be some additional things that we're going to learn about cellular respiration along the way. Eukaryotic cells do this in the mitochondria. Prokaryotic cells just kind of do it on their membrane, their plasma membrane. Speaking of the mitochondria, here it is. And so, here's our friend, the mitochondria. And we're going to look at all of the different structural elements of it. Notice the mitochondria has a double membrane. We've talked about this before. The double membrane, the outer membrane, and then the inner membrane. The inner membrane is where the business of cellular respiration is going to be happening. In fact, the space between the two membranes is very important. We're going to be talking about the inner membrane space, or the intermembrane space. The space on the inside, the matrix is also very important for this. Sometimes you'll see the folds. They're given a name called cristae or cristae, and you'll see them here, these individual folds, which allow for more surface area to volume ratio, so that more ATP can be made without increasing the volume. Mitochondria has its own DNA and ribosomes, all these things that we have talked about before. Very important to just sort of be able to recognize a mitochondria and its anatomy. So here's our general overview of cellular respiration. Cellular respiration is three reactions: glycolysis, the Krebs cycle, or on this particular picture called the citric acid cycle. We're going to use the term Krebs cycle, continuing our theme of people's names from the Calvin cycle. Those citric acid cycles probably a little bit more descriptive term, and then the electron transport chain, which is where oxidative phosphorylation and chemiosmosis take place. And so let's look at these three individual reactions. There's actually a sort of a half thing that's going on here that we're going to talk about with pyruvate oxidation as well. That is a pretty important element of this. So glycolysis, look at the word glyco. Anytime you see glyco, you're thinking sugar, right? Lysis is something that we've seen before with hydrolysis. We've talked about lysosomes, right? It has to do with breaking. And so this is literally breaking glucose. What glycolysis does is it breaks glucose down into two pyruvate molecules. A glycolysis is a 6-carbon sugar. Each pyruvate is a 3-carbon molecule, so it literally just cuts it in half. Right. As a result of that, a little bit of ATP is made. Sometimes you'll see this substrate level phosphorylation associated with it. Anytime you see the word substrate, of course, that's going to make you think enzyme, right? And so there's a lot of enzymes involved in the process of glycolysis. And I think, we have a special little slide for glycolysis, so we'll get there. And so we'll get more into that. And this pyruvate is the star of the glycolysis. This happens out in the cytosol, which is outside the mitochondria, inside the cell membrane, not in the nucleus. That's a pretty specific place, right? All living things do glycolysis. It's one of those conserved processes that link all living things together. In order for the pyruvate to get into the mitochondria, this process of pyruvate oxidation takes place. Remember, we talked about oxidation, reduction. Reduction is when you receive electrons and it reduces charge. Oxidation is getting rid of electrons. So pyruvate is going to essentially get rid of some electrons. It's going to oxidate, and it's going to oxidize. It's going to turn into something else, right? It's going to lose a carbon. We'll look closer at that in a moment because I'm pretty sure I don't have a slide about pyruvate oxidation. And so we will talk about it here, and I'll mention it a little bit more. So pyruvate's going to lose a carbon. Anytime a carbon is lost in this reaction, it is lost as CO2. CO2 is one of the byproducts, or the byproduct of this reaction. And it's given off here as one CO2. The rest of the CO2 is going to be given off in the Krebs cycle. And so this acetyl, which is just a 2-carbon molecule, is going to be then put on the Krebs cycle, right? And so pyruvate oxidation is sort of like an intermediate reaction that you do see a little bit more written about in the AP literature now. I used to just group it in with the Krebs cycle, but it's not really part of the cycle. All right, speaking of the Krebs cycle, here it is in all of its glory. This says citric acid cycle. Why does it say citric acid cycle? Well, we're going to look at that in just a moment. Purpose of the Krebs cycle is to create electron carriers. A little bit of ATP is made, but the main idea behind the Krebs cycle is to create electron carriers. The Krebs cycle happens in the matrix of the mitochondria. And then the third step is the electron transport chain. This happens on the inner membrane of the mitochondria. This is where oxidative phosphorylation occurs. Oxidative happens in the presence of oxygen. Phosphorylation is when a phosphate is added to something. Purpose of this is to use the electron carriers created in glycolysis and Krebs in order to make ATP. And we're going to talk about how that's done. Let's look at glycolysis a little deeper. So glycolysis is breaking down glucose. We have glucose there on the left. And you can see that a little bit of ATP is made. In fact, a little bit of ATP is required to break the glucose into 2 separate pieces. And so this is not as much pushed in the literature anymore, but there's this idea of spending a little bit of energy to get more energy out. And we used to talk about the investment phase and the payoff phase. Not as important anymore in the literature, so don't concern yourself so much with that. But it is okay to know that in glycolysis, a little bit of ATP is made. Some organisms are able to get away with this. Glycolysis does not need oxygen in order to function, and so some organisms live without oxygen, are able to do glycolysis and produce enough energy to allow that organism to live. Another thing that is made is NADH. NADH is an electron carrier, and don't get it confused with NADPH, which is in photosynthesis. I always tell my students that the P in NADPH stands for photosynthesis. It doesn't, but we're going to pretend that it does, because it doesn't matter what these letters stand for. We're not going to learn those. And so if there's a P there, it's in photosynthesis. If there's not, it's in cellular respiration. These electron carriers are actually really important when we talk about fermentation, very important. These electron carriers are headed to the electron transport chain. And most importantly, we have 2 pyruvates that are made here. These are 3-carbon molecules. They're headed to the Krebs cycle. Actually, they're headed to pyruvate oxidation, then to the Krebs cycle. Here's the Krebs cycle. So remember we talked about the pyruvate oxide, or the glycolysis, making 3 carbon molecules. Well, notice up here, only 2 carbons get on the Krebs cycle, right? And that's because one of those carbons has already been given up during pyruvate oxidation. And so we'll talk about that more. I don't think we'll talk about it more, actually. So there's nothing really there. We used to have to know acetyl-CoA and coenzyme A and all that stuff. It's not as important anymore. You don't see it as much in the literature. In fact, all of these steps of the Krebs cycle are pretty unimportant. As long as you understand that these 2 carbons get on and they both get off, but every time they get off as a CO2, right? And the important idea of the Krebs cycle is that these electron carriers, NADH, 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 oh, here's FADH2. FADH2 is the also ran of the Krebs cycle. It's sort of like, hey, I'm an electron carrier too, and everybody's like, yeah. It is an electron carrier. It's just not as robust as NADH. Whatever, moving on. And so these 2 carbons get on the Krebs cycle. They're going to get off of CO2, and the cycle just sort of continues, right? But these electron carriers are made. That is the big deal behind the Krebs cycle. And how many times is the Krebs cycle going to happen per glucose molecule? We make 2 pyruvates. Those pyruvates are broken down a little bit through pyruvate oxidation, and each one of them is going to get on the Krebs cycle. So Krebs cycle is going to happen twice for each glycolysis or for each glucose molecule. So let's look at the idea of fermentation, because we're going to look at cellular. We're going to look at the ETC a lot more detail in day 2. And so fermentation. First of all, why does fermentation occur? Fermentation occurs when there's no oxygen, and so glycolysis does not require oxygen in order to occur. The Krebs cycle won't happen if there's no oxygen, but it doesn't require oxygen either, right? After glycolysis, the cell makes a decision. The decision is, if is there oxygen or is there no oxygen? If there is oxygen, then Krebs cycle is going to happen, ETC is going to happen, which is over here. If there's no oxygen in the cell, then fermentation is going to happen. The purpose of fermentation is to free up electron carriers so that glycolysis can continue. All right. The purpose of fermentation is to free up electron carriers so that glycolysis can continue. What does that even mean? Let's look. So let's go back up here. In order for glycolysis to happen, one of the reactants for glycolysis is NAD+, right? And you can see that. Well, NAD+ is created when NADH goes and drops off its electrons. NADH cannot drop off its electrons at the ETC unless there's oxygen present, because oxygen is the final electron acceptor in cellular respiration. And so if there's no oxygen, NADH piles up. And if NADH is piling up, there's no NAD+ being made. And if NAD+ is not being made, there's no glycolysis happening. So fermentation is sort of an alternate pathway, sort of a detour, so that NADH can drop off its electrons someplace else, so that NAD+ can go back and feed into glycolysis, and glycolysis can continue. You can see it on both of these pathways. There are 2 kinds of fermentation. There's lactic acid fermentation, which you see on the left, and alcohol fermentation. I almost said ethanol fermentation, either way. Animals do lactic acid fermentation. All other organisms that do fermentation do alcohol fermentation. But notice the same sort of thing is going on here. What do we notice in this first step, glucose to pyruvate? This is glycolysis, right? Electron carriers are made, ATP is being made, 2 net ATP is what it says there. Without oxygen, NADH cannot drop off its electrons, and so this step, pyruvate to lactate, is what we call NAD+ regeneration, right? We're making more NAD+ so that glycolysis can continue. When it does that, of course, pyruvate is going to be changed, accepting those electrons. It becomes lactate in animals. This is lactic acid. In other organisms, like bacteria, yeast, they do this ethanol fermentation or alcohol fermentation. Same idea, though. NAD is being regenerated. In this case, CO2 is given off, and an ethanol is created, which is a 2-carbon alcohol. This is why, like when you mix yeast in with dough, the yeast can't breathe, and so it makes CO2, creates alcohol. Of course, when you bake the bread, the alcohol burns off, and the bread rises because of the CO2. This is how other alcoholic products are made through this process of fermentation. But again, the purpose of fermentation is to regenerate NAD+ so that glycolysis can continue. Let's look at a practice problem. Student performs an experiment measuring ATP production in yeast, both aerobic and anaerobic conditions. Aerobic oxygen, anaerobic no oxygen. The data below shows the data below. In aerobic respiration, average of 30 to 32 ATP are created, and in fermentation, 2 ATP are created, which the following best explains the difference in ATP yield between the 2 conditions. We know that more ATP is going to be made if that cellular respiration is allowed to continue, Krebs cycle and then ETC, whereas glycolysis is going to produce an overall less because oxygen is not available, right? So let's go find that ABCD. Fermentation increases the number of pyruvate molecules. It doesn't. Oxygen prevents ATP from being produced during glycolysis. No. Presence of oxygen allows electrons from NADH and FADH2 to be passed through the electron transport chain, leading to oxidative phosphorylation. That's correct. I don't even need to look at D. C is the correct answer. What does D say? Just for giggles. Aerobic respiration produces more glucose. Respiration doesn't produce glucose; it breaks it down. Right? So C is the correct answer. I hope this was helpful. We're going to be looking at the ETC in part 2. I hope you can join us.